Hello, good afternoon, Mr. Raman and friends. Today we are going to presentation about plant resistance against pathogens. First of all, let me introduce ourselves. Next slide. My name is Putri and Sekar as moderator. With my friends here are Kesya, Shafa, Viantari, Raden, Laura, Suzana, Dessya, Ayuda, Hamidatus Solichah, Tiara, Novita, Andriana, Maura Bennyta, and Pramono Dwi Sulistyo. Let's start the material.
Resistant development through conventional plant breeding. First, plant breeding is an activity carried out to improve plant character that are inhabited in a new population with new genetic traits. Plant breeding products are cultivars with special characteristics as desired by the breed, such as high production, tolerant to marginal environmental conditions, resistant to pests and diseases, etc. Number second is pest population with high genetic variation are important breeding material for the assembly of high yielding varieties. The basic population that has high variation will give a good response selection because high genetic variation will provide a great opportunity to get the right cross combination with a good of traits super.
There are three plant disease resistant mechanisms. Number one, mechanical resistance. Number two, chemical resistance. And number three, functional resistance. Next. Number one, mechanical resistance. Active mechanical resistance. This resistance is the result of the interaction between the genetic system of the host plant and the pathogen. This resistance occurs due to the formation of a cell layer that line the infect plant part. The formation of couplings on the cell walls, a separating layer consisting of a layer of cork, gunfeld cells, or blendok, abscess, and telosis cells. Passive mechanical resistance. Plants that have passive mechanical resistance have a morphological structure that makes it difficult to be infected by pathogens. For example, it has a waxy coating. It keeps the plant from getting wet so it avoids fungal germination. As few stomata will avoid fungal infection that penetrate through the stomata.
Next are chemical resistance. Chemical resistance is divided by two. First, active chemical resistance. This resistance is the result of genetic interaction between the pathogen and the host plant. This resistance is in the form of hypersensitivity reaction or HR in resistant plants. In resistant plants, the cells surrounding the pathogen or infected cells lose their turgor, turn brown, and the cells die immediately, thus avoiding the pathogen from spreading to other cells. In addition to the emergence of hypersensitivity reaction, plants can also detoxify or neutralize toxins produced by pathogen. And two are passive chemical resistance. A pathogen can attack a plant because the plant has cell contents which are the nutrients needed by the pathogen. Passive chemical resistance is formed because the plant does not have the nutrients needed by the pathogen. In addition to this mechanism, passive chemical resistance can also be caused by the presence of pathogen inhibiting substances such as acid, oil esters, phenolic compounds, and other substances. 3. Functional resistance. This resistance arises because of its growth, so the plant can avoid disease, even though the plant is actually susceptible. Certain pathogens usually only attack plant at certain growth phases, so that if at that growth phase, the environmental conditions do not support the growth of the pathogen, the plant will avoid pathogen infection. Plants that ripen quickly are often protected from new diseases that come at the end of crop season.
Relationship between resistant mechanism and disease resistance can divide by two, which is number one is plant resistance and pathogen virulence, and number two is pathogenic rest and plant resistance.
Plant resistance and pathogen virulence can also divide by three, which is mechanism of variation in fungi, mechanism of variation in virus and nematodes, and mechanism of variation in bacteria.
The mechanism of variation. First, in fungi, one parasitic fungi develop different mechanisms for different purposes. For example, to infect the host plant, obtain nutrients, and to colonize the tissues it has infected. During the early phase of pathogenesis, heterotrophic parasitic fungi maintain the integrity of host cells and tissues. They absorb nutrients for their respective living host cells. It can be said that the mechanism is mutation, sexual hybridization, heterokaryosis, and sexual recombination. Second, in bacteria, the emergence of genetic diversity and variation in bacteria is caused by the process of gene recombination between one type of bacteria and another. Recombination or exchange of these genes through gene transfer. Genetic material and plasmid can move through various mechanisms such as traduction, transformation, conjugation, and transposition. Then in virus and nematoda, parasitic plants and nematodes penetrate directly. Pathogens initiate attack at the rate of vegetative growth, so human spore and seeds of parasitic plants must germinate first. Viruses and mycoplasma can penetrate through wounds or be introduced by intermediaries or vector, but we have to know that bacteria, viruses, and mycoplasma never penetrate directly.
Next here is pathogenic rest and plant resistance, which is divided into two parts. That is vertical and horizontal resistance, and the two gain by gain and protein by protein hypothesis.
Vertical and horizontal resistance. Vertical resistance. This resistance is controlled by a single gene or multiple genes of a cultivar responsible for resistance. Often, this is effective only for certain pathogenic races and does not provide resistance to other races. This type of resistance is called specific resistance or vertical resistance. Horizontal resistance. Horizontal resistance is regulated by a number of genes. It is generally said that varieties that show horizontal resistance are slow disease formers. Due to the following mechanism, reducing the number of lesions produced per unit of inoculum and shortening the reproductive life of the wound. Gene by gene and protein by protein hypothesis. The genetic relationship between plants and their pathogens is not clearly known. However, some researchers conclude that for each genetic locus, there is one that regulates the resistance or sensitivity of the host, and there is a locus that regulates whether or not a pathogen is virulent. This relationship is called the gene-to-gene hypothesis, which states that each gene in the host is capable of mutating to confer resistance, and that there are genes in the pathogens that are capable of mutating to deal with the resistance. The protein for protein or protein by protein hypothesis was put forward by van der Planck, according to which the compatible relationship between the pathogens and the host is due to the presence of pathogenic proteins that match the host proteins. In susceptible plants, that pathogens excretes a protein to the host that copolymerase with the protein complementary host. In line with the gene for gene hypothesis, hosts and pathogens recognize each other through their proteins. If the plant is susceptible, copolymerization occurs while resistance occurs if there is no copolymers between the host and the pathogens.
That is the presentation by our group. If you have any question, you can ask them on discussion group on SPADA. Thank you for your attention.